Hello, in this video I'm trying to explain to you about linkage and the more complicated bits of genetics. So first of all, it's really important to understand the importance of random or independent assortment and segregation. So what that means is when the pairs of homologous chromosomes can align in any order in metaphase 1. So if we've got the spindles like this, then the chromosomes can line up like that. But each pair could be either way around, like that, okay? And in dihybrid inheritance, that means that when the genes are on two chromosomes, you'll get even numbers of all possible gametes. So, if the genotype is big A, big B, little b, on metaphase 1, the chromosomes could line up this way around like this. So you would end up with these two chromosomes going to that end, forming a big A, big B gamete, and these ones going to that end, forming a little b gamete. Or the chromosomes could align this way round, in which case they'd split down here. Those ones would go to that end, forming a little a, big B gamete, and these ones would come to that end, forming a big A, little b gamete. And that allows the formation of all of the Mendelian ratios that we've talked about. So big A, big B, little b crossed with big A, big B, little b, giving a 9:3 to 3:1 phenotype ratio. And then things like the dihybrid heterozygous test cross getting a 1:1 ratio. So what is linkage? Linkage occurs when instead of the two genes in dihybrid inheritance being on separate chromosomes, different chromosomes, they're on the same chromosome. So we call them linked. And that means that the alleles cannot segregate because they cannot separate independently as easily. So in this case here, although the genotype looks like big A, big B, little b, really the only gametes that are going to be formed are big A, big B when that chromosome goes that way, or little a, little b when that chromosome goes that way. So how do you write this down? Well, you write this down using these parallel lines, and each line represents a chromosome. So just as I had shown a second ago, if these are our two chromosomes like this, okay, one chromosome would have big A, big B, so that represents that line, and this one, would be little a, little b. It is possible to form what we call recombinants, but these will be less common than what we call the parental gametes. So in this setup here, we've got big A, big B on one chromosome, little a, little b on the other. So these gametes, the big A, big B, little b, are called the parental gametes, and you would expect to have lots of these, but you could form recombinants by the process of crossing over. So if this chromosome crossed over with that chromosome, then what you would end up with is a big A, little b gamete and a little a, big B gamete. So those are called the recombinant gametes. Now these will happen less frequently than the parental gametes, and consequently the recombinant gametes and therefore the recombinant phenotypes occur less frequently, which disrupts the expected Mendelian ratios. So a classic question for this sort of thing is in the cross big A, big B, little a, little b crossed with little a, little b, little b, what genotype would the recombinant offspring have? So what you've got to bear in mind is all of the offspring will have one gene, one gamete from that parent and one gamete from that parent. So if I think about what the recombinant offspring would look like, well, they're going to have the recombinant gametes, okay? So here a recombinant gamete would be big A, little b, and up here the recombinant gamete would be little a, little b. The other possible recombinant gamete would be little a, big B, and then little a, little b. So those guys are the recombinant offspring, and you would have fewer of those than the parental offspring, which would all be big A, big B, little b, or little a, little b, little a, little b. I hope that helps you to solve some of your genetics problems.